Assalamualaikum warahmatullahi wabarakatuh. So we continue with lecture number 2 for week number 1. So again, we want to do the revision of organic chemistry. So the chemical bond in the micromolecule. So what are the reaction that can combine molecule into macromolecule? Okay, so from one, let's say one C3 becoming C100. Okay, so we have first is the ester bond, then we have the amide bond, and then we have phosphodiester bond, and then we have glycosidic bond. So there are a lot more bond or linkage, but here is in the biomolecule, uh, important in the biomolecule. Okay, so first we see the ester bond. So ester bond is the esterification reaction, the condensation reaction of a carboxylic acid with alcohol or phenol. So these are alcohol to produce an ester and water. Okay, so these are the carbolic acid. This one carbolic acid, and these are the alcohol. So you can see that it combine OH from the carbolic acid with H from alcohol, so it produce the ester and the water. This one, the byproduct is water. So in order to make it much faster, so we need to have an enzyme, or in this case uh, acid. Okay, a strong catalyst enzyme. So sulfuric acid. So these are examples. So we have here R C double O O H, carbolic acid, and these are the alcohol. So we can produce this ester with the ester bond with the hydro water. Okay, so these are the production. Okay, so these are A, and these are B, and these are C, and these are D. Okay, so if we want to produce this, okay, or we want to increase the production of the product here, so we can, in this case, we can add more alcohol, or we can remove the product. So this reaction will go to this way, to the right. Okay, so we're going to have more ester uh, product. Okay, so this also the ester bond. Okay, so this one is from glycerol. So these are glycerol, and 3 molecule of stearic acid. Okay, so these are 3 fatty acid or stearic acid. So you can see that this one combine H and OH become H2O, and this one combine with the ester bond. This also the ester bond. This also a ester bond. Okay, so we can combine 3 R. So these are, let's say these are R1, R2, R3 to this glycerol through this ester bond. Okay, so next is the amide bond. Okay, so again the same as the ester bond. A condensation polymer is a polymer formed when the difunctional monomer are linked together via condensation reaction with the production of byproduct water molecule. Again, see here you can see that O, this OH react with this H from this amine. Okay, amine. So producing the product and the byproduct as the water. Okay, so here you can see that if you want to combine two amine, okay, so we can use this reaction. So here you can see that this N will combine with this C, making the amide bond, or in this case amide linkage. Okay, CNO, and producing the H2O. Okay, and this here you can see that this also, if we have another molecule, so you can also do the amide bond, so producing another a longer chain of polymer. Okay, so next is the phosphodiester bond. Okay, so here is between the sugar in the adjacent nucleotide. So we are going to see this later. So here we have the phosphate. Okay, phosphate and the alcohol. Okay, OH. So I'm doing the this reaction, A plus B, so become this uh, this product with the phosphodiester bond. One bond here, one bond here, and the byproduct H2O. So these are the example. Okay, so here you can see that there is two different sugar. Okay, with different this R1, this R2, but the same phosphate. Okay, so the phosphate from group number 5, then the 3 hydroxyl here, the 3. Okay, this one 3 hydroxyl OH we react with the phosphate next the molecule next to it. Okay, so these are 5 marking this bond. Okay, so we can see that O is phosphodiester bond. So this one bond and this one bond. Okay, so we have this linkage of these two sugar, making it into 2 sugar. Okay, and this one 3 at position 3 can also make another phosphodiester bond if it can happen. Okay, so making the chain longer and longer and longer. So the last one is the glycosidic bond. Okay, so these are the bond between two monosaccharide resulting from the reaction between the hemiacetal carbon OH group of the one monosaccharide and an OH group of the other monosaccharide. Okay, so here these are just alcohol, and these are the uh, let's say the sugar. Okay, so OH combine this OH plus this H producing H2O, and then we have this product HCO O and R. Okay, so these are the example. So we can see that here there is two alpha D glucose and D glucose. So this OH combined with this OH, okay, and it producing this glycosidic bond with the oxygen. Okay, so these are the oxygen. So you can making this chain, okay, a longer chain, and the byproduct is H2O again. Okay, so we are saying about this nucleoside and nucleotide before this. So what is nucleoside and what is the nucleotide? So nucleoside is a combination of a sugar and a base, and produced when the nitrogen atom in the in this case just example pyrimidine and or purine base from an N-glycosidic bond to carbon 1 of a sugar. Okay, so here these are the examples: sugar plus base, nucleoside, and the byproduct is 2O, and these are the example. So here you can see that these are the adenine and these are the ribose. Okay, so this adenine plus the ribose. Okay, so ribose is the sugar, adenine is the base. Okay, so this H plus OH. Okay, so becoming H2O, and this one making the N, so glycosidic bond, not oxygen, but this in this case the nitrogen making this bond. Okay, glycosidic. So that is the nucleoside. And next is the nucleotide. So nucleotide are nucleoside. So we have the nucleoside in which a phosphate group bond to the OH group on a carbon pi 5 of ribose of the deoxyribose. Okay, so here these are the example, the adenosine triphosphate and the guanosine 5 monophosphate GMP. So AMP. So here you can see that this one, these are the nucleoside. So when we have the phosphate group combined to this H5 or carbon 5, so it become the nucleotide. So next is we have this AMP. So we have ADP and we have ATP. Okay, so here we have this ATP and the energy. So adenosine triphosphate ATP is storing the function is to store and transport the energy in its high energy phosphate bond to the place in the cell where energy is needed. Okay, so we have this molecule so going around our body, and when anywhere of our body need the energy, let's say the muscle. Okay, so this one will uh, do the hydrolysis and producing the energy. Okay, so without uh, the without uh, changing the carbohydrate. Okay, so because the carbohydrate is our source of energy, but this one is the main, not the main, the fast way to get the energy. Okay, so here hydrolysis. So you have see that from the ATP can become the ADP, producing the phosphate ion, the H ion, and also 35 kilojoule energy. Okay, so this 35 kilo energy can be uh, given to our muscle or our protein to do the work. Okay, temporary work. So these are hydrolysis and these are the synthesis. This way is synthesis, this way is hydrolysis. Okay, so next we see the biological macromolecules. So we have polysaccharide, protein, and nucleic acid. Okay, so these three are built out by these elementary entities. So polysaccharide from monosaccharide, protein from amino acid, and nucleic acid from nucleotide. Okay, and linked by generous ester bond. So it can be longer and longer and longer chain or bond chain. Okay, so first we see the polysaccharide. Okay, so a complex carbohydrate in which 10, 100 or even 1000 of simple sugar are linked together through glycoside bond. Okay, so here you can see that these are monosaccharide. So if we have the enzyme, it still is a monosaccharide. So because this is the smallest entity of this saccharide. Okay, disaccharide. So we can see the enzyme. So we can cut this bond, become two monosaccharide. So polysaccharide, if we enzyme, so we have many monosaccharide molecule. Okay, so these are the polysaccharide. Okay, so the polysaccharide can be uh, from the branching. Okay, so here these are unbranched chain homopolysaccharide. So the chain is continue. So these are 1, 1, 1, 1, 1, 1, 1. So these are unbranched, no branching of polysaccharide. Okay, so here you can see that this one we have a straight line or a continuous chain of monosaccharide. Okay, this one we have a branch chain homopolysaccharide. Okay, so meaning that the isomer or the configuration. Okay, so you can see here these are the branching point. So these are the branching points. So there is a lot of monosaccharide with a branch. Okay, so here you can see that, and these are the same, the same monosaccharide. So next is the unbranched chain heteropolysaccharide. So here we have two type of monosaccharide. Okay, so this one blue, this one is red. So different R. Let's say these are R1 and this one is R2 or adenosine. So we have repeated this chain. Okay, a long chain, unbranched chain, but a different monosaccharide. Okay, and next is the protein. Okay, so protein are the polymers of amino acid linked together chain by amide bond, and the small protein are called polypeptide. So the amide bond or amide linkage also known as peptide bond. So these are the protein and their function. So some of the protein are used for the structural, provide structure to our body or our cell. So contractile, so making the muscle move. Transport, so carry some substance through our body. Storage, store the nutrient. Okay, hormone, regulate body metabolism. Enzyme, so some protein can be enzyme, so speed up the biochemical reaction. And then some protection, so just like our white blood cell. Okay, so here, so these are the example of the protein. The next one or the last one is the nucleic acid. So there are linear polymer of nucleotide. Okay, we see linked by a phosphodiester bond. So we have two type of nucleic acid. So the first one is deoxyribonucleic acid, DNA. So the function of DNA is to store and transfer the genetic information. So let's say from father to daughter to son. Okay, so we have the same, nearly the same genetic information. Okay, so next one is the ribonucleic acid. So the function is to synthesize the protein, to produce the protein, and okay, so it can produce many type of protein. So the protein that we're saying, this one in this case, the molecule that carry out essential cellular function. Okay, so it can, by combining so many conjugation, so it can become the, let's say, structural, transport, storage protein. Okay, by this one. Okay, so these are the structure of nucleic acid or the DNA. So phosphate, sugar, phosphate, sugar, phosphate, sugar. So for DNA, backbone, so deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose, and RNA, phosphate, ribose, phosphate, ribose, phosphate, ribose. Okay. So these are the example of DNA and RNA. So you can see that DNA is the alpha helix. So let's say here we have the adenine, the R functional group or R group. So R So here red is adenine, yellow is thymine, blue is guanine, uh, green is cytosine, and purple is uracil. So the amino acid, okay, the amino acid. So you combine together, making this alpha helix. So it becomes this DNA. For RNA, is just one long chain. Okay, so that does not making the alpha helix. Okay, so here these are the protein. Okay, so when RNA, let's say, making this bond, okay, so it can produce the protein. Okay, the protein. Okay, so these are the base. You can see here sugar phosphate backbone. This one phosphate, ribose, phosphate, ribose, phosphate, ribose, and these are the, here you can see that these are the amino acid that is uh, connected to the uh, deoxyribose or the ribose. Okay, so that is the sum of DNA and RNA. So thank you.